about the Pisces, and Pisces includes the fishes. The topics that are mentioned in the syllabus about the Pisces are respiration, locomotion, and the migration. And if you will see the previous year questions, here the questions have been asked about the respiratory organs like accessory respiratory organs, or even in the base, how there is this aerial respiration, how the gill respiration takes place, the account of gills, accessory respiratory organs. So we will discuss them in detail. The locomotion in fishes is important. The types of scales have been asked, the muscles and fins have been asked, and about the flying fishes has been asked, even about the bony fishes, the scales in bony fishes. There have been questions about the role of fins in locomotion. We will discuss these in detail. The migration is an important topic, and the question have been asked about the types of migration, that is anadromous and catadromous. We will explain this in detail. So starting with the general characteristic of the fishes or Pisces, you will see that in generally you will observe there is a streamlined body. There are different types of fins like caudal fin, there is anal fin, there is pelvic fin, there is pectoral fin, there are these dorsal fins, there is a lateral line, there is operculum in order to cover the gills. So these are the general features of the fishes. Talking about the general features, the fishes, the habitat of the fishes, they are aquatic, they are freshwater and marine both. So they are aquatic and can be both marine and freshwater. The body structure of the fishes, they are streamlined. Okay, they are streamlined, and the body, it is divided into head, trunk and tail, and the body is covered by scales. There are different types of scales. We will study them in detail. And the locomotion is using the fins, whether through the pectoral or pelvic fins, or even due to the dorsal, caudal or anal fins. The tail fin is also called as a caudal fin, and it helps in the propulsion. The endoskeleton is made up of either cartilage in case of chondrichthyes, or bone in case of the osteichthyes. The respiration is through gills, and the gills, they are covered by the operculum. In case of chondrichthyes, there is no operculum. The circulatory system is closed. There is two-chambered heart, that is, it contains one atrium and one ventricle. The circulation is single circulation, that is, the blood, it will pass through the heart only once. There is excretory organs. These excretory organs, these are mesonephric. They excrete ammonia. That is why they are ammonotelic, and also urea in case of marine. In case of marine, there is urea, and in case of freshwater, there is ammonia. The nervous system contains brain and sensory organs. There is lateral line system for detecting the vibration and movement in the water. The reproduction is oviparous or ovoviviparous or even viviparous, and fertilization is external in both mostly bony fishes, and internal in case of the cartilaginous fishes. So the development can be direct or indirect. And the temperature regulation, there is they are cold-blooded organisms, that is the poikilothermic. They cannot regulate their own body temperature, so it varies according to environment. If the water is cold, their body will be cold. The swim bladder in most of the bony fishes, they have the swim bladder in order to maintain the buoyancy, so that they can float in water, and it's absent in the chondrichthyes. The mouth position is ventral in cartilaginous fishes, and it's terminal or subterminal in the bony fishes. And mostly the fertilization is external in osteichthyes and internal in chondrichthyes, and sexes are separate. The male and female will be separate. The major classes of the Pisces include the chondrichthyes and the osteichthyes. So you need to know about them. In case of chondrichthyes, they live in the marine environment. The exoskeleton contains placoid scales. The endoskeleton contains cartilaginous skeleton. There is amphistyle type of the jaw suspensorium or jaw suspension, and the gills act as a respiratory organ. The operculum is absent, and the caudal fin, there is heterocircle caudal fin, that is, the caudal fin, it is not equally sized, it is unequal. And then the male have claspers, that are the reproductive organs, and they do not have air bladders. For example, scoliodon. If you will see the structure of the scoliodon, there will be presence of ventral mouth. There are these gill slits. There is no operculum. There is pectoral fin, pelvic fin. There is anal fin. There is a caudal fin. There is a second dorsal fin and first dorsal fin. So this is a diagram of the cartilaginous fish or the dogfish, that is scoliodon. And in case of osteichthyes, these are marine. These can be freshwater. They can be brackish. They have bony exoskeleton. That's why osteichthyes. The exoskeleton consists of ganoid, chasmoid, ctenoid type of the scales, and there is jaw suspension. They have autostylic jaw suspension, and they do not have claspers. The gills have operculum and air bladders are present, for example lepidosiren. And this osteichthyes, they have subclasses, that is actinopterygi and sarcopterygi. Actinopterygi contains the ray-finned fishes and sarcopterygi contain lobe-finned fishes. So this is just an overview about the different types of fishes. Now discussing about the respiration. So respiration in fishes, it has evolved in a manner in order to accommodate with the growing need of oxygen requirement in the body. So there was this evolution of respiratory system in manner that there was development of multicellular complex organisms with time, as the hierarchy and the evolution kept on happening. There was increase in the complexity, increase in the efficiency, as well as oxygen requirement, because there was increase in the reutilization of energy. So there was also this increase in utilization of energy, and you know that the energy is basically the ATP, these ATPs, and these ATPs are generated using oxygen. So that is why the respiratory system evolved, and there are 4 principles of respiratory system evolution. Why it evolved: in order to provide an extensive surface area for gaseous exchange, for example alveoli in lungs and lamellae in gills. It also reducing the energy cost and development of single path of respiration rather than different paths. Then it also depends upon the permeability of the membrane. So the better the permeability, better is the exchange of gases. The cutaneous respiration is not as efficient as it is in the alveoli and gills. So in order to maintain the continuous breathing to produce a continuous supply of energy in the form of ATP. So this is why the respiration evolved. So talking about the respiration, first we will see the respiration in cyclostomata, for example petromyzon or the lampreys. So they mostly have a parasitic lifestyle. The general body structure of the lampreys is this way, and these are the external gill apertures. So they have gill pouches inside them, and there are gill openings. So there will be gill pouches and gill openings. There are about 7 pairs of gill pouches. Then there is this buccal chamber. This is a mouth. This is a velum which acts like a valve, and there are pharyngeal muscles. So the mechanism is that there will be tidal ventilation. Firstly, the tidal ventilation, it means that the water will enter and exit from the same pore. So there is this bidirectional flow of water. So this is called as the tidal ventilation. Next is the buccal pumping. Buccal pumping means that this buccal chamber, it will act like a pump, and there will be coordinated movement between the gill pouches and the mouth. So the water will go in and then it will go into the gill pouches, and from there will be exchange of the gases. The buccal pumping includes the water will enter into the mouth, then to the buccal chamber, then through the velum it will enter the gill slits, and then it will come out of the gill pores. So this is about the respiration in cyclostomata. Now we'll discuss about the respiration in the myxine or the hagfish. Here you will observe that there is this mouth from where the water will enter, and there is also nasal opening, and this is the pharyngeal portion. There are efferent and afferent ducts attached to the gill pouches. There are 5 to 16 gill pouches and a common opening. The pharyngeal cutaneous ducts are present. So the gill pouches are around 5 to 16. There are gill openings, and internally the gills contains filaments or the lamellae. The mechanism is that the tidal ventilation, it's bidirectional flow. As I told that there is tidal ventilation. So from the same opening it will enter and go out of the body. The mouth and the nasopharyngeal duct also get involved in the respiration. The water will enter in the gill pouches and the pharynx, and then there will be exchange of gas. The muscular pumping also takes place, that is through the muscle contraction and relaxation. So this is how the myxine respires. Then about the respiration in the chondrichthyes. If you closely see the structure of the chondrichthyes, you will observe that there is one common mouth open. When the mouth opens, there are different gill lamellae. These are the gill lamellae. These a type of gill lamellae are called as a holobranch gill lamellae. Holobranch gills. And so you see this here we have hemibranch, that is the lamellae are present only on one side. So there are different gill slits, and these remain open, and there are here it is shown that open gill slits, here the gill slits remain closed. So there are gill arches, there are branchial pouches, there are these gill lamellae where the exchange of gases takes place. This is pharynx and this is esophagus. So this is a general structure of the respiratory organs of chondrichthyes. The respiratory organs contain about 5 to 7 pairs of the gill slits, and they are located laterally behind the head. So this is the head, and behind the head there are these gill slits. The gill arches support the gill filaments, which are actual respiratory surfaces. These gill structures have gill arches which have two rows of gill filaments. They can be hemibranch and they can be holobranch. The filaments are rich in the blood capillaries for the gas exchange, and the gill lamellae, it increases the surface area. Then what happens next, you will see that there is no operculum. So the gill slits, they open directly to the outside, and there are also spiracles. These are the small openings behind the eyes, and it is a vestigial first gill slit. So these spiracles are the vestigial first gill slits, and they just help in drawing in the water, especially when the mouth is buried. So the water flow mechanism we have two main mechanisms. First is a ram ventilation. In case of the ram ventilation, in active swimmers like sharks, the water will enter through the mouth and it will exit through the gill slits. So this is a ram ventilation. The water will enter into the mouth and it will exit through the gills. And second is the buccal pumping. In buccal pumping, the buccal cavity pumps the water over the gills even when it's stationary, and it's less active species. It is in the less active species. Ram ventilation is the major type of ventilation which happens, and you will see that the sharks and the chondrichthyes, they are continuously moving in water. The reason being they need to have this continuous flow of the water over the gills. Then next is this gas exchange. The gas exchange occurs over the capillaries. There is highly vascularized capillaries over the gills, and there will be this exchange of gases, that is carbon dioxide and water, and there is this counter flow mechanism between the water, and it even maximizes the oxygen uptake. So this is how the respiration in chondrichthyes takes place. Next is about the modern bony fishes. In modern bony fishes, you will observe something like the same mechanism, but there will be operculum present. There will be, this is the gill arch, these are the gill arches, and you will see these are the gill filaments, and these are highly vascularized, so that there will be easy exchange of the gases. So the respiratory organs include 4 pairs of gills. They are located in the pharyngeal region, and each gill is supported by a gill arch. So each gill is supported by, this is the gill arch. And then these lamellae, these are rich in the capillaries for efficient gas exchange. There is operculum, I told you about it, and then the mechanism is, firstly it will intake on inhalation through the mouth. The fish will open the mouth and lower the floor of the buccal cavity. It will create a negative pressure when the water will enter into the buccal cavity, and due to the negative pressure, the water will flow into the mouth, and during the exhalation, the buccal cavity, the pressure will increase and the buccal cavity will push the water outside. The operculum, it opens slightly and the water is forced over the gill filament, then out through the opercular slit. and this results in the continuous unidirectional flow of the water over the gills for efficient gas exchange. There is also this counter current exchange mechanism. This water and blood, water and blood, they will flow in the opposite direction. The water and blood will flow in the opposite direction across the gill lamellae. There will be gill lamellae. It will create a concentration gradient. It will allow maximum oxygen diffusion into the blood and carbon dioxide removal from the blood. There is also a swim bladder. Swim bladder is not a respiratory organ, but it helps in the buoyancy regulation. It is found in the Lepidosteus, amia, which are the primitive teleost. It can function as a lung-like organ or physotomous condition. So there are also external gills. These external gills, these are very rare in adult fishes, but they can be found in the embryonic or the larval levels. These are highly vascularized outgrowths for gaseous exchange. Here is an example. This is an external gill. Okay. So in case of lungfishes, you will observe it. For example, the larvae of the Protopterus, Lepidosiren, Neoceratodus, they have the external gills. They are used for respiration. Then you will also observe in some of the embryos of the sharks and rays, there will be filaments which arise from the gill arches. There will be presence of Polypterus larva where you will observe the external gills, and the adaptive significance is that they increase the surface area for gas exchange in low oxygen environments, for example swamps and stagnant water, and they are also useful during the stages when opercular and internal gill slits are, or the gill system is underdeveloped, and during that time there is respiration through the external gills. You will also see that in some cases, for example amphibia, there is this neoteny, so the larval traits, they will be retained as external gills in the adults too. Now we will discuss about different types of the accessory respiratory organs. Firstly, we have cutaneous respiration, that is through the skin. It is common in the eels and mud skippers. Then buccopharyngeal epithelium. In case of clarias and heteropneustes, you will see here the clarias and heteropneust, you see this is a suprabranchial cavity. These are these arborescent organs, and there are first gill arch, gill filaments, gill fans. So this suprabranchial cavity, it acts as a organ for respiration. The lining of the mouth and the pharynx is highly vascularized, and this fish, it will gulp in the air and absorb the oxygen through this lining. Next is the intestine or the rectum. Some fishes even swallow the air and absorb the oxygen via the intestinal lining, and they expel the used air through the anus. It is seen in the catfish or loaches. The swim bladder in physostomous fishes, the swim bladder, it connects to the gut and acts as a lung. It is seen in the lungfish. And then labyrinthine organ, it's a highly vascularized bony structure above the gills in some fishes. For example, anabus, that is the climbing perch. There is also the suprabranchial chamber found in the clarias, which is highly vascularized. You can see in here there is this portion called as a gill chamber that is found as amphibians. You will observe in the kana there is this gills and suprabranchial cavity. There is labyrinthine organ. You can make it in case of anabus. In case of this heteropneust, you see that here is the gill chamber and even the tubular, large scale and often seasonal movements from one habitat to another for the purpose such as for spawning, for feeding, or for avoiding the unfavorable conditions. The migration, they can be daily, they can be seasonal, they can be lifetime events, and they involve the movement from the freshwater to marine to freshwater, or from brackish to freshwater. So this is how the migration takes place from one place to another. So migration can be for the breeding purpose, that is to produce the offspring. For example, the salmon and the hilsa, they will migrate in order to produce the eggs or lay the eggs. Then next is feeding. The fishes, they will move from a nutrient scarce area to a nutrient rich area in order to feed, where the food is abundant and it ensures the growth and survival. For example, in case of tuna and sardine. Then next is in order to avoid the unfavorable conditions. The migration helps the fishes in case of the extreme temperatures, droughts or pollution. If one area has extreme drought like situation or pollution, they will move to another area where there is favorable conditions for the growth of the fishes. Next is the growth and development. The growth and development for of the juveniles, it is in order to move the juvenile to a place where the growth and development can take place easily, and they will migrate to the habitats which are more favorable for growth and development. Next, they will also move to the areas of where there is oxygen availability. There are areas which are not oxygen, which are eutrophic. For example, some eutrophic region. So these fishes, they will move to the oxygen rich areas, and in order to avoid the predators, also to run away from the predators, to save their lives, they will move from one place to another. For example, there are these juvenile reef fishes. They will move in order to avoid the predators. Then we have different types of the migration based on the movement from freshwater or the marine or the salt water concentration. For example, we have anadromous fishes. Anadromous means the fish, it is born in the freshwater and they will migrate towards the sea. They will migrate to the sea and they will return back to the freshwater in order to breed. For example, salmon. The catadromous fishes are those fishes which are born in the sea and they will migrate to freshwater for growth and they will return back to the sea in order to breed. So it is opposite of anadromous. For example, anguilla, we will study them in detail. Then amphidromous means that they will move between the freshwater and saltwater but not for breeding, for other purposes. For example, galaxias. Potadromous are those fishes which are entirely within the freshwater systems. So they migrate within the there is this freshwater system and they will migrate within this whole one system. For example, there is Katla, there is Labiorohita. Then oceanodromous, that is they will move or migrate within the marine system only. For example, sardines, tuna and herrings. So the types of migration based on the habitat, first one was the let's see, there is this potadromous. It is between freshwater, for example, rainbow trout. Then oceanodromous, in the ocean only, sea to sea for mackerel. Then sea to freshwater, it is anadromous, and catadromous is freshwater to sea. And amphidromous means which keeps on doing, oscillates between the seawater and the freshwater. So this image is from the book Mastering Zoology in 89 Days. You can get this book. It's a very good book and you will see the comprehensive coverage of the syllabus through these flow charts. So you can get this book. It's available online. Next, there is this anadromous migration. This anadromous migration, in this case, the anadromous fishes like salmon and hilsa, they undergo several morphological, physiological and behavioral changes to adapt from the marine, that is salt water, to the freshwater environments during the migration for spawning. So what happens if you look at the life cycle of this salmon in detail? First, there will be spawning, and this Atlantic salmon, they are born in the freshwater streams, and they will where the adult, this salmon will return to spawn, and spawning typically occurs in the same area where the salmon were born, a behavior that is known as the homing. So after this spawning, the eggs and the 11th stage, these eggs will be deposited. These female salmon, they will deposit the eggs in a nest or red. So the nest is also called as the red. So this will be the nest. They will remain inside the nest and they will produce the eggs. The eggs are fertilized by the male salmon and they develop into elvins. So elvin will develop, or the par stage. Par, they will inhabit the freshwater habitats for a period of time, for a long period of time, and then there will be small smaltification. In the smaltification, there will be changes, physiological processes, there will be transformation in the body morphology, behavior, and this is called as a smaltification, and it occurs during the spring. There will even be the change in the physiology, change in the color, and then they will migrate to the ocean after the completion of the smaltification. The young salmon, now they are called as a smalts. They will migrate downstream from the freshwater to the oceans, and where in the oceans they will even navigate through the estuaries, coastal waters, and they will slowly adapt and they will slowly grow in the marine phase. In marine phase, they will spend several years feeding and growing in marine habitats, and then what happens? They roam in the North Atlantic Ocean from the coastal waters of North America to the waters of the Greenland, and during this phase they will diet on small fishes, crustaceans, and then there will be this return migration. Return migration is that they will again be seen going back to the place, going back to the area, and they will utilize the Earth's magnetic field and even the navigational cues, even the hormonal changes. Then they will reach in their natal rivers, and the adult salmon, they will spawn in the same manner as their parents did, and they will complete the life cycle there, and they will even die there. So in the anadromous fishes, the changes in the body, there will be osmoregulatory changes. That is, the fish will now adapt to the marine water and even to the fresh water. It happens through the changes in the gills, kidneys, the changes in the retaining of the salts or removal of the salts from the body. It will also involve the change in the control by the cortisol and prolactin. There will be hormonal changes, that is increase in the cortisol. There will be surge in gonadotrophin, testosterone, and even the gonadal development of prolactin will help in the adaptation to the fresh water. There will be gonadal maturation. The gonads, they will enlarge significantly, and energy is diverted from the growth and maintenance towards the reproductive development. Then there will be metabolic and energy shifts, that is increased metabolism to fuel the long distance swimming. There will be use of stored fat and muscle proteins. There will be no feeding during the freshwater phase. The muscle and the cardiovascular adaptations include that there will be increased heart efficiency and increase in the red blood cells. There will also be the color change. There will be change in color, and the morphological changes will be in the form of, there will be hooked jaws, there will be enlarged teeth, there will be mating rituals and competition. The behavioral changes, for example, strong homing instincts using the geomagnetic cues, and increased aggression will be observed, and territoriality during the spawning. So this is how the anadromous migration takes place. In case of the catadromous migration, again you will see there will be this is observed in the anguilla, that is eel. Firstly, the spawning takes place. This spawning takes place in the Sargasso Sea, that is the north region of the North Atlantic Ocean. The after the spawning, the larvae, which are called as a leptocephalus or leptospalus, they are very transparent and ribbon like and very narrow. They feed on the marine snow and planktons. They will arrive at the coastal waters, and here they will undergo metamorphosis, and they will later on migrate to the fresh water, and in the fresh water they will grow, they will feed there, and here there will be growth and maturation in the fresh water, and later they will again migrate to the Sargasso Sea, where they will do the spawning and they will die there. Similarly, they will also have the changes in the body, for example, hormonal changes. Similarly, gonadotrophin, thyroid hormone, cortisol, they will increase. There will be stimulation of the gonadal development and osmoregulatory shifts. Gonadal maturation will take place once they will reach into the Sargasso Sea. Once the leptocephali will reach into the saltwater ocean again, there will be osmoregulatory adaptations like hyperosmotic conditions, hypoosmotic conditions. In order to fit in these conditions, they will also change the mechanism of salt storage in the body, or the gill will start to excrete the excess salt in order to preserve the hyperosmotic or the hypoosmotic conditions, or transition or create a transition between the two phases. There will be increased energy storage, lipid and glycogen reserves will be there. There will be change in the color and morphological changes. For example, coloration will become silvery and metallic, and the eyes, they will enlarge and skin will thicken. There is this lateral line system, it becomes more developed, and there will be behavioral changes. For example, strong homing instinct, they have this strong homing, they want to go back to the home, that is home. They will have this strong homing instinct, the process where the animal, it is it has a strong drive to go back to home. They will migrate thousands of kilometers to reach the home, and they will become nocturnal, and they will migrate in large schools. For example, European eel or the anguilla does this. They will migrate for 6,000, more than 6,000 kilometers to the Sargasso Sea. Just they move through the water using the muscle contraction, body flexion, and the fin movement, and it's primarily adapted for the swimming. Though some fishes, they can also move across the mud or even land, but primarily fishes are found in water, so the locomotion will be in the form of swimming. There are various forces that act on the body of the fishes which create a drag against the body. So there are various forces against the body. There are these forces or drag against the body of the fish, and this fish has to move, has to do the locomotion in the opposite direction of this drag, in the opposite direction of this force. Firstly is the viscous drag. Or the frictional drag. So it happens due to the viscosity of the water, and it occurs due to the friction between the layers of water and the fish body. So if this is the fish body, this is the fish body, there will be a proper viscous drag. The viscous drag will be due to the layer of the water and the fish's body, and it increases. This viscous drag will increase due to the surface area of the body, swimming speed, and water viscosity, and it's minimized by the smooth skin, scale modification, mucus secretion, and slim streamlined body. For example, the sharks, they have dermal denticles that reduce the boundary turbulence and viscous drag. Then next is the pressure drag. This pressure drag, it is the form drag. Okay, so this pressure drag, it is caused by the shape of the fish body. It results from the pressure between the front and the back of the body as the water will flow across it. So for example, this is your fish, and then there will be water will move across the body. It will create a pressure on the body. So this is the pressure drag, and it is minimized by the streamlined or fusiform body and tapered head and tail. And you will see that the tuna and swordfish, they have streamlined bodies in order to reduce the pressure drag. Then is the wave drag. Wave drag is when there is when the fish is near the water surface. For example, this is the water surface, and there are waves across the water surface. This fish, it will face drag due to the waves, due to the water currents. There are ripple waves. It is minimized by the swimming deeper below the water. The more this fish will spin deeper in the water, there will be less wave drag because the water is calmer in the lower layers. There is one induced drag, that is with the production of the lift or thrust from the fins and the tail. It is caused by the vortices and turbulent flow around the moving fins. So it increases with the fin size and motion. The bigger the fins, the bigger will be the creation of the induced drag. So these are the different forces against which the fish has to move. There are different types of forces involved in the fish movement. First one is the thrust, that is the forward motion. It propels the fish. Then there is drag, that is the backward motion. It is the resisting motion. and it must be overcome. Then is the lift motion, that is upward motion. It maintains the elevation in the water. Then is the buoyancy, in order to balance the weight and prevents the sinking. Then the weight is also a type of force. It acts due to the mass and it must be countered. There is reactive force, that is opposite of water push. It enables the forward motion of the body and it follows the third law of motion. To every action there is opposite reaction. Then we have the role of different fins. The fishes, they have different types of fins. For example, we have these caudal fins. This is at the rear end, at the tail end, and its primary role is the propulsion of the organism, and it generates the thrust in order to move forward. There are these pectoral fins. These are just behind the, they are present just behind the, for example, this is our fish. This is the caudal fin. Here we have pectoral fins. It helps in steering and turning. It helps in braking. It is used to slow the speed propulsion. Then we have pelvic fins somewhere here. Pelvic fins are just ventral and below the pectorals, and they help in balance and stabilization, and it assists in the vertical positioning. There are also dorsal fins. The dorsal fins are the midline on the back. They help in stability and prevents the rolling during the swimming. Then anal fins. Anal fins are present on the ventral side of the anus. Again, they helps in the stabilization and it controls the side to side movement. There are adipose fins. These adipose fins are behind the dorsal fins and they help in the stability or sensing the flow. The modification in the fins, they are due to different reasons. First, if you will see the modification in the fishes for the for swimming, firstly you will see, observe, there is this tail fin morphology. Depending upon the environment, there are different types of tail fin, caudal fin. It can be homocircle, it can be heterocircle, so there are different types of tail fin morphology. The body of the fishes, it's streamlined body. So streamlined body, it helps in the fishing and it avoids the drag. The muscle arrangement. There are presence of myotomes, and these myotomes, these are mostly W shaped in the fishes in order to provide an arrangement which helps it to easily swim. Then next is the presence of the swim bladder. Swim bladder is there to maintain the buoyancy. For the buoyancy, there is a proper sensory system to notice the water flow. There is mucus coating on the body. There are respiratory adaptations. So these are different modifications in the fish's fin system in order to swim. Then there are different types of the locomotion. First one is the anguilliform motion. You see there are these undulating motions. It includes the body undulations in the wave-like motion. It's called as the anguilliform motion. It's very highly flexible and it shows maneuverability. For example, eels and lampreys. Next is the subcarangiform motion. In subcarangiform motion, there is half undulations. The half of the body shows these undulating motions. For example, trout and salmon. Then carangiform. In carangiform, only the last third portion of the body will show the undulation. The rest of the body will be stiff. For example, mackerel and jack. Then cuneiform motion. In cuneiform motion, only the caudal portion will act as a oscillating or the undulating organ, for example tuna and shark. Then ostraciform motion. In ostraciform motion, it's only the tail fin that will oscillate and rest of the body it's rigid. For example, pufferfish and boxfishes. These are poor swimmers. They often use fins for fine movements. Then there is use of median and paired fin for locomotion. For example, there is ragiform motion. In ragiform motion, there are large pectoral fins. They will undulate like the wings and they will help in the gliding. For example, in case of the stingray, they show the ragiform motion. Next is the duodontiform. They use the undulation of the broad pectoral fins. They are precise and slow movement. For example, porcupine fish. Next is the amiform. Amiform is seen in the bowfin and seahorses. There are long undulating dorsal fin for slow swimming. So there is this amiform for slow swimming dorsal fin. Next is the gymnotiform, and there is this ventral fin in case of the gymnotiform, for example knife fish. And then ballistiform. Ballistiform is the use of the dorsal and anal fin in the undulation, for example triggerfish. These caudal fins, there are different types of caudal fins too. For example, we have homocircle where upper and lower portion are equal, for example rohu. In heterocircle, upper lobe is larger, for example shark. The hypocircle means that the lower lobe is larger. Anapsids, these anapsids are extinct. Dificircle, it is symmetrical and fused caudal fin, for example lungfishes or coelacanths. The Gephyrocircle, there are no real lobes, they are straight. At the end there will be straight, for example ocean sunfish. Here you will see there is this Proterocircle or Dificircle. There will be complete fusion at the end. There will be it will be symmetrical and fused. Then we have heterocircle. There is difference in the lobes. This is hypocircle. The lower one is larger. Homocircle means where they are equal. Then we have homocircle in this case also. And here this is the Gephyrocircle where there is no real lobe. Next, you will see that the fishes have different types of scales. This scale, if you see here, this is the placoid scale. It has a median spine, and these are the flanges of the of these placoid scales. This is a ganoid scale. There will be a nucleus in between, and around this there will be concentric lines of growth. These are cycloid scale. In cycloid, they will be circular in shape, and this is the ctenoid. In ctenoid, there will be a canal opened, and then there will be these lines of growth like this, and they have teeth at one end. This is called as a ctenoid. So the types of scales, there are different types of scales. The scales form the exoskeleton of the fish. It has dual function, that is defense as well as locomotion. It decreases the drag. So the first is the placoid scale. It is found in only the cartilaginous fishes. These are tooth-like. They have a pulp cavity, dentine and enamel, and they do not grow with the fish. Nerve scales are added as the fish grows. They give rough texture or sandpaper texture. For example, sharks raise skates. They help in order to reduce the drag. Next is the cosmoid scales. The cosmoid scales are the primitive and found in the extinct fishes. And these are made of cosmine, that is a dentine-like substance, and they have enamel and bone layers. They are thick and heavy, and they are not flexible. They are not found in the modern fishes. Then we have ganoid scales. These are found in primitive bony fishes, for example, gar, bichir, sturgeon. Then these are diamond shaped. Then we have cycloid. These are found in soft ray teleos, that is modern bony fishes. These are circular or ovoid, the common scales that we see, for example, in rohu, salmon and carp. Then we have ctenoid scales. These are spiny rayed, and these aid in reducing the friction. They are similar to the cycloid scales, but they have a tiny comb-like projection called as a teni. This comb-like projection is